Dr. Yeager, thank you very much for your presentation. We really appreciate you being here with us. And now please let me introduce our next speaker, who's Professor Diego Bonilla from DBSS. Welcome.
Hi, everyone. I'm glad to be here again with you just to share some data for this convergent functional genomic analysis that we performed to identify some potential biological networks that could mediate and explain what are those mechanisms behind the creatine supplementation. So before I started, I just would like to point out this a nice review with some other ideas of the opportunities in sports nutrition. And basically, it covers several of the historical methodologies that has been used to evaluate some of these effects or physiological responses, metabolic changes after given nutritional or exercise strategies. But interestingly, in the last years, the bioinformatics, the omic technologies, and some other platforms that offer a multisystemic and systems biology-based point of view has been raised. So in here, we have known that the central dogma of molecular biology is basically explained as a fluent change between the DNA, RNA, and the proteins. But nowadays, we know that this is a little more complicated because even proteins can also go and regulate as transcription factors or any other kind of proteins, the DNA replication. And also, we know that the epigenomic and epigenetics part of the regulation of DNA involves methylation, acetylation, and even lactylation. It has been seen in the few years. So the microRNAs, I won't skip it because actually those are very important to regulate the gene expression. And all of these panorama just take us into these kind of omics that are multi-integrative sciences that try to understand a given biological entity by a group. Not only in a single and low throughput analysis, but trying to identify patterns, to doing sequencing, and to identify some applications for medicine, pharmaceutical industry, or agriculture. Because it's very interesting to find that we are 99.9% identical, but the thing that makes us different is just resides in very specific sections of our genes. And if we take these data to across the species, we will find some kind of convergent properties as well, where some species, as we, the humans, with the mice, and with the frogs and some other, we have certain patterns and sections of our DNA that are shared. And actually, the most distant species could have a 14% of homology in our sequences, which is very impressive. So taking advantage of all of these advances that have been done by evolutionary biology and phylogenetics and this stuff is where we came with this kind of omic approach, where we try to analyze the all genes at the same moment and as a group, or we can use a transcriptome or transcriptomics analysis, or proteomics or metabolomics, as we were seeing in the last talk by Dr. Jagger, where the metabolites can be analyzed as a group or individually. So what is very important here to clarify is that every complex system, as the cells or a tissue or a system or our own body, could have these emergent properties that are some special characteristics that are arising after the interaction of the given nodes and entities that are inside this huge system. And that importantly, they are not part of these individual components of the systems. So that's why we have this beautiful scenario where the bioinformatics starts working hard with large amount of data to do these kind of things, alignments, data integration, data visualization, workflow modeling, in order to understand how these large amount of proteins or genes or transcripts, they interact with each other. So we have currently in the different associations, the National Library of Medicine, it has covered with the NIH a lot of these databases, but the European side, they have also the EVI, the EVI, the European Bioinformatics Institute, and some others that just take us to nice and very curated, that is manually doing by experts in biology, data that is being deposited in these repositories in order to offer these kind of secondary or an open source analysis for the researchers. So that's why we came to this kind of approach, where we try to collect the data that was published in these repositories using the NCBI and the EVI. And after that, we rerun the differentially expressed genes and make it this convergent analysis with the mice. So finally, we got some set of differentially expressed genes that converge between these species, and we run an upstream regulatory analysis for pathway reconstruction. We found that 3 data only has been published using mice and two from mice and one from a human. That has been using this kind of transcriptomics analysis and technology to collect what are the genes that are changing after creatine administration. So the 3 sets of data were one from a clinical trial by Safdar and colleagues, and the other 2 were tissue with animal in vivo, and the other one was in vitro. So what is very interesting here is that this set of genes that we found that they can be differentially expressed, they have a similar pattern in some of those functionalities that we can see it here after the enrichment analysis. Actually, what we do is that we take this set of differentially expressed genes. It is aligned with a set of libraries that exist in the literature with these bioinformatic tools. And we got a set of tables that are ranked by differentially significance and the network topology. So we found that the gene ontology, there is an effort to standardize and classify what are the uses and the applications of these genes, their characteristics. So we find that in the biological process, the creatine network that is changing these kind of differentially expressed genes and are so important for neurodifferentiation, for the regulation of angiogenesis, the mammalian gland development, and some others. And also the cellular component and molecular function just take us to the DNA regulation. So that's why we found that the transcriptomics analysis in here, in the upper left side, we find this set of transcription factors that potentially regulate the network, and those interact in here in the B. You can see it with some intermediate proteins that are just seen in the literature as well, and experimental evidence for these bioinformatic tools. And finally, they take us in this pathway reconstruction to the kinases, the main secondary messengers that regulate the network from a given receptor activation. So in here, that's why we came with this kind of network and signaling pathway that explains some of these different steps. Actually, you will find in much more detail in our paper, but we find that at the first moments of the creatine supplementation that it is start increasing, so there is an increase in phosphocreatine levels, as we already know. And this is regulating by several other experimental evidence that's back this kind of importance activation of the AKT and the PKB protein. We have seen also reports of the myostatin reduction in human training with resistance training. And there has been also reports of activations in the P38 pathway And the MAP kinase is a very important MAP kinase that regulates cellular differentiation, and some others that exist right now in the literature. So you will find this literature verification, and which is interesting of this approach is that it saves a lot of time when you are trying to go specifically to a given protein. Actually, Dr. Ostojic recently published something about the clotho protein, and there are some molecular mechanisms using oocytes that are the most frequently used technique and type of cells to use this patch clamp analysis for voltage changes in the transport of different molecules. And the clotho has been shown that it can increases and stabilize the creatine transporter, at least in vitro, in these oocytes. So there are other things that are very interesting, and is that there is a sort of regulation that is depending on the concentration of the creatine in the cell. So once the creatine starts increasing, it reaches a top. So we know that it's like 150 or 160 millimoles of creatine per kg of dry mass, of muscle dry mass. So in this case, some of these important regulations that we found is that there is a pathway with the TXNIP. There is a thyroxine regulating protein that can use this kind of endocytosis of the creatine transporter, just to decrease and control, or probably the amount of creatine that is increasing in the cell. So this actually was found to be increased, this gene and all of this pathway, in one of the the animal models that we analyzed. So we here just put a lot of information that exists in the current literature, some other potential mechanisms that are used with these question marks, and some others that could explain the activation or a given pathway. So it's very nice, comprehensive, with literature and experimental evidence that we are citing in this paper. And also, we perform also microRNA detecting analysis, just to see if there are some common microRNAs that can be upregulated or that regulate the network. And it's very interesting to find that these kind of elevation in these kind of microRNAs has been associated with some of the molecular effects and the physiological effects that we have found throughout the conference. For example, the cancer regulation, also the immune response, and the neuroprotective effects. So perhaps there are some hidden and also intricate molecules, especially microRNAs, that can regulate all of these physiological effects that we see after creatine supplementation. So this is the paper. So I would like you to take a look, just to have a clear image, and just to say that the science and all of these fractal models are evolving, and we probably in the future we could try to modulate and to use this kind of artificial intelligence to predict the behavior of biological systems. So in this case, what I'm showing is something very impressive that just I'm learning to this in a second master that I'm doing. And actually this leaf just was constructed using some very simple mathematical functions. So just iteration, and it gave us to these fractal models. And this is the way that we try to understand systems biology as well, from the interaction of simple nodes that just are repetitive. So we hope that we continue being ready for all these future challenges that are coming from. And thank you so much.